Great, thank you so much. It's a pleasure to participate at the Gatsby Plant Summer Science School this year. So a huge thank you to the organizers and for the attendees for making this possible this year. And actually, it brings me memories of the first Gatsby Plant Summer Science School that I attended. That was in 2006, and I was a PhD student, and we were asked to help with some of the lab practicals. So this is the overview of my talk. I'm going to give a brief introduction on light signaling and then how I became interested in photobiology. And then I'm going to talk about signal integration in the nucleus, in nuclear hubs, in order to regulate plant adaptation. And we use an Arabidopsis as a model system. And I'm going to talk about two physiological processes, hypocotyl elongation and flowering time, as a readout. And then I'm going to end with some conclusions, future questions, and potential applications. So thanks to the amazing program, the great talks and workshops that you already had as part of the summer school, I'm sure you are aware that plants are extraordinary organisms. So apart from the fact that they have to adapt to changing environmental conditions, they also have to make their own food, right? They have to make their own energy because they're photoautotrophs. So light is essential not just for energy production through photosynthesis, but light also acts as a very important informational signal to control plant development, morphology, and also adaptation. And we need to keep in mind that light doesn't work in isolation. It works with other environmental and endogenous stimuli to shape plant development and morphology. So this is just an example of many physiological processes that are regulated by light throughout the life cycle of the plant. So from germination to deethylation or photomorphogenesis, which is where the plants emerge from soil and see light for the first time, to the production of natural sunscreen that accumulates on the epidermis of plants, photoprotective compounds. Also, light controls stomatal behavior, and trains circadian rhythms, and also the duration of light, of day length within a 24-hour period is very important for determining whether a plant will transition from the vegetative to the reproductive phase, which means it will stop making leaves and it will start making flowers. And also light quality as well as quantity or intensity is very important for informing the plant whether it's under a dense canopy and it needs to escape from it in order to capture more light. So obviously light is really, really important in plants. And that's why they have evolved very, very different photoreceptor systems compared to animal rhodopsin-based visual systems. So in Arabidopsis and in plants in general, there's 5 main photoreceptor families. And starting from the shorter wavelength, which is also the more harmful because it has the higher energy content, UVB is perceived by the UVB receptor, which is UVR8. And in blue light, there's 3 different families of blue light receptors that perceive this wavelength. There's the cryptochromes, the phototropins, and the Zeitlupe family. And red and far-red light is perceived by phytochromes. There's 5 phytochromes in Arabidopsis, Phi-A to Phi-E. Now, photosensing happens through the specific domains in each photoreceptor family, with the exception of UVR8 that senses UVB through its tryptophans. All the other photoreceptors have photosensing domains that attach to a chromophore. So in the case of blue light receptors, the chromophore is primarily flavin-based. In the case of phytochromes, the chromophore is phytochromobilin. Now, all these photoreceptors need to integrate all these different light stimuli in order to shape plant development in response to changing environmental conditions. And I would like to draw your attention to the fact that the majority of these photoreceptors, with the exception of phototropins that are plasma membrane-associated proteins, all the other photoreceptors are either localized or translocate into the nucleus in order to signal. So basically the action is happening in the nucleus. And it's known that in response to light, there's major transcriptional reprogramming that takes place, especially during photomorphogenesis, which is when plants perceive light for the first time. So all these photoreceptor families are able to integrate signals in the nucleus to induce hypocotyl elongation, shade avoidance, photoprotection, flowering, clock entrainment, depending again on the developmental stage and the environmental conditions. Now, it's not surprising that sunlight goes hand in hand with an increase in temperature. And in recent years, climate change obviously leads to increase in global temperatures. And a lot of studies have shown the temperature increase of a little as 1 degree Celsius can lead to huge crop losses. So this is, of course, species specific, and some species may benefit from an increase in temperature, but the trajectory suggests that there's going to be a huge issue in food production with increase in global temperature. So this is something that is really important to understand in more detail. And since light and temperature go hand in hand, it was not very surprising when recent studies showed that a lot of light signaling components are shared by ambient temperature sensing pathways. So in particular, the photoreceptor, the red light receptor, phytochrome B, not only can perceive red light, but it also acts as a thermosensor. And other signaling components such as ELF3, early flowering 3, which is a component of the clock, but also of light signaling, has also been shown to act as a thermosensor. So the plant basically uses common signaling components to induce adaptive responses to light and also temperature. So in response to elevated temperature, so we're talking like a transition from 22 to 28 or even 32 degrees, not heat shock, just a higher temperature, we can see that shade avoidance and high ambient temperature induce very similar changes in plant architecture. And this results from changes in gene expression. And again, these genes are shared. So some of these changes in morphology is elongation of the embryonic stem, the hypocotyl that you've already heard about, and also of leaf morphology, so they become more epinastic, and also initiation of flowering. So all these responses in terms of thermomorphogenesis are important for avoiding loss of water, basically. Now, how I became interested in photobiology. So originally I come from a small island in Greece. There's plenty of sunlight. We usually try to hide from it. But it was only when I moved to Scotland for my undergraduate degree that I realized the importance of sunlight, possibly due to the absence of it. But actually, we had really amazing lectures as an undergraduate student in molecular biology that were on photosynthesis, but also on light signaling. And I think this is what attracted me to plant biology per se. And that's one of the reasons that I attended a guest seminar by the late Professor Winslow Briggs. And he was talking about this novel photoreceptor family that was identified in his lab. There were blue light receptors, the phototropins, and they were they're controlling phototropism, which is directional growth towards light. So this phenomenon was reported by Darwin, and it was only in the late 1990s that the gene was cloned and identified, which is amazing. So in his talk, he showed, genetics, the genetic screen, the physiology, and also hardcore biophysics and photochemistry of how this protein absorbs light and induces signal transduction. And I thought that's amazing. You can, combine all these approaches to study one protein in one lab. So at the same time, I had the opportunity to apply for a work placement program, which is basically you can do 1 year as part of your degree research in a lab. And I thought it would be amazing if I could go to this lab to work on a project like this. And I did. I emailed Winslow. And thanks to a Sainsbury undergraduate studentship, this made possible. I was able to go to Stanford to do my work placement, and that was my first introduction in terms of research, and it was on phototropins, the blue light receptors. So during that time, I was able to present some of my work at an international photobiology conference, and I met a lot of scientists, and I heard about different photoreceptors, and I thought that's the area that I really want to continue for my postgraduate studies. And at the time, in Glasgow, at the lab of Professor Gareth Jenkins, there was a gene recently identified that was important for photoprotection. So we didn't know what the protein exactly was doing. We knew it was regulating gene expression, but at the time, there was no known UVB receptor in nature in general. So it was a great opportunity to work on this protein and understand how it functions. Later, it was shown through collaborations that it is actually the UVB receptor. And while I was applying for postdoctoral fellowships, I had the opportunity again through a Gatsby-supported postdoc to Professor John Christie to study a different aspect of photobiology. So we were using the light sensing domains of these blue light receptors, and we were using them as fluorescent reporters that had some advantages over GFP. So they were smaller in size, so you could tag viral proteins and monitor their spread in Berthemiana. So again, that was really exciting because you could see how you can use knowledge from plant photoreceptors and apply them to science that can be used by different areas in biology. So for my postdoc, I really wanted to go back to the US. So thanks to a Human Frontiers Fellowship, I was able to join a lab that I was admiring very much, the lab of Professor Joanne Corey. And there I became more familiar with signal integration and actually the importance of thinking of the big picture. It's not just one wavelength, one stimulus. It's the big picture, different stimuli, environmental and endogenous, and they all regulate plant developments. So I was fortunate enough to be allowed to take part of my project to start my own lab at Glasgow. And the model system we use is Arabidopsis. We study light-induced responses, so photomorphogenesis, flowering initiation, and also crosstalk between different wavelengths and also with temperature. And we use a specific protein as an example of controlling these responses. And ultimately, we want to understand, how specific proteins can redirect the balance between growing and adapting, in this case, growing or flowering, for example. We use different approaches, molecular biology, so next generation sequencing. We use cell biology, so imaging, and also physiology and genetics. So when I started working on this protein, we knew that it regulates hypocotyl elongation in response to blue light, and we knew that it can regulate gene expression. However, we didn't know how this protein functions. So one of the ways to understand, what your protein of interest may be doing is by having a look at its amino acid sequence, because there is a lot of important information there. So you can use the sequence and you can put it through different software predictive sites, and it shows you if there's any specific motifs or domains that can indicate its function. In the case of this protein that got its name by its domain composition, tandem zinc finger +3 or TZP, it has an extended N terminus that is predicted to be unstructured or intrinsically disordered. And then towards the C terminus, there's 3 domains, 2 zinc finger and a +3 domain. So zinc finger and +3 domains have been shown to be involved in transcriptional regulation and chromatin remodeling. The combination of these domains is unique to TZP and to this protein basically in nature. So that was something very interesting. So we knew that this protein regulates hypocotyl elongation. We knew that it wasn't a canonical transcription factor, but we wanted to know how it controls gene expression. So one of the things we did is to try to identify interacting partners. So from that, we identified from that screen a transcription factor that is named zinc finger homeodomain 10. And we showed that these 2 proteins interact directly. And through genetics and next generation sequencing, we showed that they can associate with specific promoters of genes that are known to be very important for hypocotyl elongation. We know that these 2 proteins are part of a complex that act downstream of the photoreceptors, right? So this is one of the ways this protein, TZP, can induce gene expression that regulates hypocotyl elongation in response to low blue light. Now, when we started working on this protein, we tagged it with a fluorescent reporter, GFP, to see where it's localized within the cell. So it wasn't a surprise that we saw it in the nucleus, since it regulates gene expression. But what we found really interesting is that it could form these nuclear bodies. And we did some dynamic studies, fluorescent recovery after photobleaching. So you can bleach one of these nuclear bodies and monitor how fast it recovers. And it's really, really fast, which suggests that possibly something is actively recruiting TZP into these parts of the nucleus. Now, you need to keep in mind the nucleus doesn't contain membrane-bound organelles. there isn't the ER around it, but there's no membrane-bound organelles within the nucleus. So that's something, very interesting in terms of how is it working. We knew it wasn't an artifact of overexpression because even when there was very low protein levels, you could still see these structures forming. We did some super resolution imaging, and we could see that these nuclear bodies are actually like donut shaped, so they're hollow in the middle. And again, we were, very interested to see what is happening there. So what is known in terms of nuclear bodies in general? So as I mentioned, the nucleus doesn't have membrane components inside it. So one way to form these nuclear bodies is through liquid-liquid phase separation. So it's basically a reversible process. You can see when you mix oil and water or oil and vinegar, and you have separation into two liquid phases, a dense and a dilute. In the case of the nucleus, you probably have a dense phase that is made of proteins and or nucleic acids, and then the dilute phase, that is the nucleoplasm. Now, it's common for proteins that contain these intrinsically disordered regions to form these biomolecular condensates or nuclear bodies, and they're regulated by different stimuli, concentration, temperature, or pH. And sometimes you have scaffold proteins that recruit other proteins into there. Now, nuclear bodies are not new. They were identified in 1903 by scientists and also artists, as we heard from George Bassett's talk, Ramon y Cajal. And later it was shown that disruption of these patterns correlate with genetic diseases, aging, and cancer in mammals. So this is a way for the nucleus to compartmentalize specific processes such as transcription, storage, splicing, changes in nuclear architecture, protein activity, and so basically there's a collection of proteins or molecules that accumulate to specific domains of, specific function. Now, in terms of nuclear bodies and plants, they're less characterized. However, there's already known components of light signaling components that can form these nuclear bodies. And in the case of TZP, we wanted to see which wavelength was inducing the formation of nuclear bodies. And we showed that it was primarily red light. And we also showed that the photoreceptor phytochrome B that was already well known to form nuclear bodies in response to red light was co-localizing with TZP, as well as L3, another component that is involved in light signaling. And we also showed by genetic studies that phytochrome B is essential for recruiting TZP into these nuclear bodies. In terms of the physiological significance, we use pharmacological studies as well as, again, transcriptional assays and genetics, and we showed that this recruitment of TZP to nuclear bodies induces the expression of flowering locus T, which is essential for initiating flowering in response to a long day photo period. So that's, again, another example where the same protein, TZP, interacts with different proteins in a different context to induce the expression of different genes. In this case, they're involved in initiating flowering. Now, as I mentioned, there is a lot of light components that are used or shared for temperature sensing or temperature signaling, thermomorphogenesis. So in the case of our protein of interest, TZP was directly interacting with Phi-B, which is also a thermosensor, and also other labs had shown that TZP is part of a multi-protein complex that, again, a lot of these components have been shown to play major roles in thermomorphogenesis as well as light signaling. So, of course, the obvious question and the hypothesis was perhaps TZP could also modulate, may play a role in thermomorphogenesis. And this is just to remind you that plants respond to elevated temperature by elongating their hypocotyls and by initiating flowering, for example. So we examined these two responses in plants that lacked, so mutants for TZP or overexpressing TZP. And in our case, we could see that TZP mutants were hypersensitive to temperature compared to wild type, whereas when you overexpress TZP, you could get a hyposensitive response to high temperatures, suggesting that actually TZP is a negative regulator of thermomorphogenesis, which again is quite surprising that depending on the stimulus or the context, TZP can act differently. So in response to low blue light, it positively regulates hypocotyl elongation, whereas in response to elevated temperature is a negative regulator, which is very interesting. So basically, again, you can see a single protein is part of different multi-protein complexes and regulates different biological processes. So depending on the context, whether it's tissue, wavelength, developmental stage, or stimulus, the output or the function of the protein may change. And again, it all depends on whether a plant needs to grow further or adapt to the changing environment in order to survive. So of course, there's so many different questions that we still want to answer, not just in terms of the protein of our interest, but in general. Who is in this? Who else is there in these nuclear bodies? The nuclear bodies that form in response to light and temperature. Is it only proteins and nucleic acids? How do they form? What are the dynamics of their formation? And also, what is their function, right? Is this the site where proteins become post-translationally modified? Is this the site of active gene expression, of transcription, as we previously showed for one aspect of TZP? And what's their impact in general in biology? Could they be affecting genome-wide nuclear architecture? That it has been suggested that light and temperature can do this. So now there are approaches that we can actually take a snapshot of what happens in the nucleus. So chromatin is like a spaghetti structure, right? It's not a linear structure. It's compacted in a way that it can fit into the nucleus. So there are techniques such as chromatin conformation capture in order to examine intra and interchromosomal interactions. And you can have these maps, contact maps, and see what happens, for example, in response to a light or temperature stimulus. You can look into different mutants and you can get maps like this that tell you what happens within the nucleus in 3 dimensions. So conclusions, very simple. We use as an example one protein and we show that it can have many functions depending on the context. It's very important to understand the details of molecular processes, but also there's physiological significance, so the big picture. And in terms of the importance, why are we interested in these processes? Ideally, we want to transfer knowledge from what we learn from Arabidopsis and photoreceptors and signal integration in the nucleus to try to understand the molecular mechanisms conferring plant adaptation in other species as well. And ideally, we want to try to optimize light capture and also plant adaptation without compromising crop yield, because this is very important, because there's always this balance. So understanding, the details of these pathways and how they're regulated can help us perhaps use them for precision agriculture through genome editing, for example, or even custom farming systems like the use of agrivoltaics. So with that, I would like to thank past and present members of my team, collaborators, funding, of course, and also my postdoc mentor, Professor Joanne Corey, and the Gatsby, not just because I'm invited to give a talk, but because they've been supporting my career, from the very, very, very early stages, and primarily through mentoring and through networking. So I'm really, really grateful to them. And with that, I hope you had lots of fun attending the summer school. I would say, plant science is fun. I'm sure you've realized that through all the amazing sessions that you had. If you're interested, find a theme that you're passionate about and follow it. You've realized that by talking to different people and also the career talks you had this morning, that every career path is different. So try to shape your path based on your own personality, circumstances, and preferences, right? Try to identify one good mentor and also a supportive peer network, which I'm sure you all have now, again, through the Gatsby, and also try to learn, from experts, whether it's a technique, and also share your knowledge and findings with the community. And the most important thing is believe in yourself and never let anyone discourage you or doubt you. So yeah, with that, thank you very much. It's been a pleasure being part of the Gatsby community.